I would like to welcome everyone to today's webinar, hosted by the GenTAC Alliance and Marfan Foundation. It is an honor for me to host this webinar with Dr. Alan Doherty, Associate Vice President for Research at the University of Kentucky and Editor-in-Chief of Arteriosclerosis, Thrombosis, and Vascular Biology. I would like to start by welcoming Michael Weimer, President and CEO of the Marfan Foundation, to say a few words.
Thanks, Mary. Good afternoon, everyone. Thank you for coming. Just a couple words, really. First of all, the Marfan Foundation is honored to be the new home of the GenTAC Alliance. We love the opportunities and the engagement, and as I'm sure you know, many of our physicians and scientists are also part of the foundation. They're part of GenTAC. The other thing is that I just wanted to mention we now have, as part of the Marfan family, we have the Hal Dietz Foundation. Came over as of July 1, and we're also 1 year in now on the vEDS movement, Vascular EDS, as a division as well. So lots of things to be excited about. Don't want to take any of the program time. Honored to be here with Hal and Mary and Alan. And with that, Mary, back to you.
Thank you, Mr. Weimer, as well as the GenTAC Alliance and Marfan Foundation for this incredible opportunity to bring together a distinguished group of scientists internationally. We welcome all participants today to type questions into the chat box throughout the presentation. Dr. Doherty and I will do our best to have each question addressed at the end of Dr. Dietz's presentation. It is now my distinct pleasure to introduce Dr. Dietz, the Victor A. McCusick Professor of Medicine and Genetics at Johns Hopkins, Director of the Smilo Center for Marfan Syndrome Research, and Howard Hughes Investigator. 30 years ago, utilizing samples from an extended family in Eastern Kentucky, Dr. Dietz was the first to identify that the locus causing Marfan syndrome was on chromosome 15, which he subsequently identified as the FBN1 gene. He has dedicated his life to the service of people with Marfan syndrome and related disorders ever since, which led to his discovery of Loeys-Dietz syndrome. Among many honors, he has been awarded the Antoine Marfan Award from the National Marfan Foundation, Colonel Harlan Sanders Lifetime Achievement Award in Medical Genetics from the American College of Medical Genetics, Research Achievement Award from the American Heart Association, and Mentorship Award from the American Society of Human Genetics. With such a distinguished research career, it is fitting that Dr. Dietz has been inducted into the National Academy of Sciences. The impact that he has made on the quality of life of people with Marfan syndrome and related disorders is difficult to measure and quantify, but to this, I can personally testify. I now welcome Dr. Dietz to share with us his experience in mouse backgrounds and genetic modification of vascular connective tissue disorders.
All right. Hello, everyone. I am going to try to share my slides. This should work, I think. I would like to thank Mary for that very kind introduction. I would like to thank the Foundation and GenTAC for the opportunity to share some of our work today. Just as a quick adjustment to Mary's introduction, I would be remiss without acknowledging the essential contributions of Lena Peltonen and Lynn Sakai and Keiko Ramirez and David Hollister and all sorts of other people who were very influential and essential in the knowledge that FBN1 gene mutations cause Marfan syndrome. Today, I'm going to talk about mouse backgrounds and genetic modification. A little bit of the message from this talk relates to the fact that there can be complications if you don't pay attention to mouse background, and I'll illustrate that a bit. But more so, I want to emphasize that there is opportunity in using differences between mouse backgrounds to take an unbiased look at disease pathogenesis, modification mechanisms, and treatment strategies. So I hope that the message that you'll come away with today is that please pay attention to background, but also use differences to your advantage. For some reason, my slide will not advance. Let's see, there we go. So this slide illustrates how mouse strains are named. I'm gonna discuss throughout the talk today two predominant strains, one called C57 Black 6J, abbreviated Black 6, and the other 129 S6 SVEV TAC, which will be abbreviated 129. Now, the name tells us a lot. The C57 Black, for example, is describing the parental strain that was created by C.C. Little in 1921. This is a very widely used strain, in fact, the most widely used experimental mouse strain. There are many congenics available, meaning different strains of these mice or subpopulations of these mice that only differ at one locus. Black 6 mice are easy to breed. They tend to be robust, and they also have a fully sequenced genome. The other strain, 129 S6 SVEV TAC, was created by Dunn in 1928. He actually borrowed mice from mouse breeding fanciers in England, and by report bred them to a strain of chinchilla. They are widely used for targeted mutations because of their excellent performance of embryonic stem cells, and they too have been fully sequenced. But you can see in these names, there's additional information. The green underlined part is substrain. The purple underlined part tells you the source. So J stands for Jackson Lab. TAC would be for Taconic. Let's look into that a little bit more carefully by looking at two highly related strains of Black 6 mice. The C57 Black 6J, which I have already described for you, and the C57 Black 6N. Now, the 6N strain was separated from 6J at NIH, hence the letter N, in 1951. These strains are highly, highly related, remain very, very similar. In fact, they're only known to be distinguished by 5 single nucleotide polymorphism differences. And also the 6N substrain lacks deletion of the NNT gene, which is inherent to the Black 6J strain. So you might think, well, really, does it matter to pay a difference to whether this is from Jackson Lab or the NIH? There was a very recent article that looked at the influence of angiotensin II infusion in these two strains. And the researchers found a dramatic difference. So at the same dose, same age, if you infuse angiotensin II into the 6N strain, they are largely protected from aneurysm, as shown by a small abdominal aortic dimension and a frequency of aneurysm of only 7.1%. However, if you do the exact same experiment with the 6J strain, they develop very large aneurysms with a frequency of aneurysms of 76.5%. So truly make a huge difference. So we're going to be describing some of our experience looking at strain differences. And initially, I'm going to talk about Marfan syndrome, which is a phenotype that I'm sure I don't have to introduce for this audience. Important point here is the very high predisposition for aneurysm and tear of the aortic root. And as we know, this condition is caused by primary heterozygous mutations in the gene encoding fibrillin-1. I'm also not going to go into a lot of detail about this slide, but over the past 25 years or so, we've used a dedicated series of what I would call hypothesis-constrained experiments to kind of chip away at the pathogenic sequence. And over that time, we've learned about important interactions or relationships between angiotensin II signaling, TGF-beta signaling, and activation of PLC, IP3, protein kinase C, ERK signaling cascade. We also know that other factors like oxytocin signaling can feed into this pathway. And we've learned that there are a number of provocations that can worsen disease that are shown in red, and other manipulations that have therapeutic benefits shown in blue. But I'd say perhaps about 4 or 5 years ago, we wondered if there was perhaps a way to take a fresh look at this in an unbiased fashion, to try to learn something new that doesn't rely upon a derivative hypothesis. So around that time, we had the good fortune of partnering with Julie DeBacher and Bart Loeys. And we and they had seen a number of families that really, with Marfan syndrome, that showed striking discrete intrafamilial variability in disease severity. And when I say that, I mean that some of the people with the underlying typical FBN1 mutation had classic and severe disease requiring surgery or experiencing dissection at a relatively young age. And other family members with the identical underlying mutation had very mild, in fact, often no vascular disease, even in older age. So we hypothesized that there might be a genetic modifier at work that influences vascular disease severity in these families. And we undertook an effort to map a modifier gene. We realized that if each family had a different modifier, that we would be unlikely to have sufficient power to find anything. But fortunately, all the families were segregating variation on human chromosome 6 that accounted for this difference in disease severity. This reached a significant LOD score, so it was a genome-wide significant result. There are 32 genes that were in this interval, but one of them kind of jumped out at us, a gene called MAP3K4. It encodes a mitogen-activated protein kinase that activates proteins like ERK, P38, and JNK. So there was a plausible mechanism by which it might relate to disease, to vascular disease. We also were able to learn that MAP3K4 is highly expressed in the aortic wall. And using dermal fibroblasts from patients with either mild Marfan syndrome or severe Marfan syndrome, we were able to show that more severe disease significantly associated with higher levels of expression of MAP3K4. Now, we didn't have the ability to recruit more of these remarkable families in an efficient manner. So we turned to FBN mice, our C1038G substitution Marfan mouse model that we and others have used extensively. And we simply disrupted one copy of the MAP3K4 gene and saw that this was sufficient to fully normalize aortic root growth rate in Marfan mice. We found that the Marfan mice had high activation of the signaling pathways that we had previously described. And again, that simple disruption of one copy of the MAP3K4 gene was able to normalize these cellular signaling abnormalities. So highly suggestive that this is a locus capable of modifying both cellular signaling and the aortic phenotype. Now, around this time, Jeff Doyle in the lab had begun to look for differences between different mouse backgrounds. So he bred the Marfan mutation onto what we refer to as an ultra 6 black 6 background, meaning greater than 20 back crosses to a pure black 6 mice. Or to an ultra pure SV129 background. And again, over 20 generations at the beginning of this study, now likely close to 100 generations of back crossing. And what Jeff observed was really quite striking. So in the black 6 background, the aorta essentially appeared normal. Here I'm showing you a long axis view of the aortic root. This is a Marfan mouse on the black 6 background. Yellow arrowheads are showing you the aortic root. We are in systole, the aortic valve is open, and the red arrowheads are showing you the more distal ascending aorta. This was very slightly bigger than wild type mice, but not dramatically so. And these black 6 ultra pure Marfan mice never went on to demonstrate aortic dissection or vascular death. In contrast, if you put the same mutation on an ultra pure 129 background, we saw a truly giant enhancement of aortic root size and growth, again shown by the yellow arrowheads and also shown quantitatively to the left. These mice did show early onset of death due to aortic dissection. We couldn't reconcile these strain-specific differences by differences in heart rate or blood pressure. Now, an interesting observation that we'll get back to in a bit is that the protected black 6 mice showed what appeared to be a somewhat generous caliber of the ascending aorta compared to the protected 129 mice, which showed a smaller dimension of the ascending aorta. But this never manifested as a fusiform aneurysm. It was always a diffuse enlargement that extended across the entire aortic arch and in fact into the descending aorta. So we would not define this as an aneurysm. As I mentioned, it never led to any vascular consequence in black 6 mice, but it was an observation that we and others have made. So here I'm showing you the aortic phenotype in a bit more of a granular way, and we're separating females from males. So as I mentioned, the black 6 mice at 2 months of age have very mild enlargement of the aortic root compared to wild type animals. There's no difference between males and females. The 129 Marfan mice have significantly larger aortic roots even at 2 months of age. And again, there's no sex specific difference. If we look at 6 months of age, you can see that there is a slight worsening of the aortic root phenotype in male black 6 mice, but still very modest sized aortas compared to their 129 counterparts that again have very large aortas and no difference between the genders. And if you look at aortic root growth, it's has pretty much parallels what aortic size has shown. Again, the 129 mice of both sexes are showing the worst performance. If you look at the more distal ascending aorta, again, the black 6 mice have more generous measurements. The male black 6 mice specifically show an acceleration in the growth of the entire ascending, transverse and descending aorta, compared to the 129 mice who show normal measurements and no sex-specific difference in aortic size or aortic growth. We looked at other aspects of the phenotype from a systemic perspective, and we're interested to observe that the 129 mice that showed a worse aortic root phenotype also showed worse developmental emphysema, as measured by mean linear intercept or distance between alveolar walls. We also found that there was slight but significant accentuation of the skeletal phenotype on the 129 background compared to Black 6, here by measuring the severity of kyphosis in these animals. So we returned to the aortic root, and we looked at the cellular signaling cascades that had been implicated through the years in predisposition for aortic root aneurysm. As you can see, the 129 highly predisposed animals show unequivocal accentuation of all of the relevant signaling cascades, including increased activation of SMAD2 and SMAD3, mediators of canonical SMAD-dependent TGF-beta signaling. Also very elevated activation of P38, protein kinase C, MEK, the kinase that activates ERK, and also a dramatic elevation of ERK activation, which we think is a distal mediator of aneurysm disease. Whereas on the highly protected Black 6 background, all of these signaling abnormalities were essentially muted. Again, this is after breeding to a pure Black 6 background greater than 20 times. So fully in keeping with the hypothesis that these abnormal signaling events are driving disease in the predisposed 129 mice. To address this further, we asked in this highly predisposed strain, do interventions that had proven protective in the past also protect the 129 Marfan mice? And the answer was dramatically yes. Treatment with angiotensin receptor blockers fully normalized aortic root growth rate, even in the severe 129 Marfan mice. And this happened with a clear attenuation of SMAD and ERK signaling. A MEK inhibitor that simply prevents ERK activation was also dramatically protective and associated with a suppression of distal signaling events, but not SMAD activation, which we believe is proximal to ERK. So what we did is we bred the two strains together. We bred Black 6 and 129 mice together for many intercrosses in order to generate extensive recombination between the chromosomes. Then we bred in the Marfan mutation and stratified the mice on the basis of the size of their aorta. So any mouse that had a small aorta, we would segregate as Black 6-like. Any mouse that had a huge aorta, we would segregate as 129-like. And the rare mice that had intermediate size aortas, we simply excluded from the analysis. And then we asked which regions of which chromosomes were consistently segregating from the protected Black 6 mice to the protected mixed offspring. And the answer was that you could explain this dramatic protection based upon the contribution of only two loci, one on mouse chromosome 5 and the other on mouse chromosome 11. Both of these loci reached genome-wide significance based upon the number of markers that were used to map this protective genetic architecture. And we found that if you consider both loci in combination, there was strong evidence that there was gene-gene interaction or crosstalk because the LOD score stating that this was a highly significant event that's likely to explain phenotypic observations jumped up to 12 if you integrated the concept of gene-gene interaction or epistasis. So this is one of the most fascinating and puzzling results that I've seen in my career. What we then did is we took mixed mice, and all we did was stratify them by their dosage of 129 alleles at these two loci of interest. So if a mouse had both alleles that were derived from the 129 parental strain at both loci, they had huge aortas just like the pure 129 mice. If mice had all alleles from the black 6 parental strain, none from the predisposed 129 strain, they behaved just like the ultra pure black 6 mice. But it was in the middle where you saw something very interesting. So if you had just one protective black 6 allele at the chromosome 5 locus, you began to see mice that had small aortas. If you increased the representation of protective black 6 alleles, you saw a higher probability of having a small aorta as opposed to a large aorta. And if you reached a high level of protective black 6 alleles, you only saw small aortas. But the interesting observation is that the representation of protective alleles was influencing the probability of having a big versus small aorta, but not leading to intermediate aortic size phenotype. So this is something that we've thought a tremendous amount about. We considered that it might be due to gender, but you can see that males and females represented as dark green or light green symbols respectively are equally distributed in the big aortas and the small aortas. We considered things like parent of origin effects. We considered things like genes in linkage disequilibrium. And we have yet to come up with a fully satisfying explanation for this probabilistic relationship between protective alleles and aortic size, something that we might return to at the end of the talk. So when we looked at these two loci, chromosome 5 and chromosome 11, there were two strong candidate genes, one called MMP17 on chromosome 5 and the other MAP2K6 on chromosome 11. We considered them strong candidate genes because there was strain-specific sequence variation that was predicted to be consequential, that it would influence the function of the respective proteins. And both of these genes were shown to be highly expressed in the aorta. When we thought about these even more carefully, it was interesting that MMP17 had previously been shown to regulate ERK activation, and that the strain-specific difference was really quite dramatic. The stop codon in one strain, in the black 6 strain, was changed to a tryptophan in the 129 strain, leading to an extension of the protein at its carboxy terminus. MAP2K6 on chromosome 11 was exciting because, again, it activates mitogen-activated protein kinases, but perhaps even more importantly, it's activated by MAP3K4, what we had identified as our strong candidate as the human modifier. And again, there's a sequence variation that was predicted to be very consequential for the activity of MAP2K6. If we looked at expression of these two genes in the 129 background, either wild type or Marfan syndrome, there was higher expression in the predisposed 129 animals compared to black 6 animals. If we looked at MMP17, again, the same message that there was higher expression of the protein as a consistent finding. So then what we decided to do was to selectively knock out each allele of both of these genes to observe the effect on aortic phenotype. So here we've bred in null alleles, targeted alleles for both MMP17 and MAP2K6. Any plus allele was selected to be a fully functional, high expressing 129 allele. And again, you can see that if you have 4 functional 129 alleles, 2 of each gene, your aorta is large, just like the pure 129 mice. If you have no functional alleles, all of them knocked out for these 2 genes, and that's all you selected for, you can see that the aortas behave just like the fully protected black 6 mice. And now you see more of the expected type of distribution of intermediate size aortas as you sequentially knock out more and more of these 2 alleles for these 2 genes. If you look at signaling, again, the results are not subtle at all. So as I've shown you, pure 129 Marfan mice have high levels of ERK activation, PKC activation, P38 activation, and SMAD activation. If you look at mixed mice that are selected for having 129 alleles at just these 2 loci, you see the identical pattern and level of activation of all of these signaling intermediates. If you just knock out both MMP17 alleles, but leave the MAP2K6 alleles intact, you see an intermediate level of signaling. And if you knock out all alleles for both of these genes, and again, just these 2 genes, you see that there is dramatic suppression of all of these signaling events. Here, this is a completely separate biologic replicate experiment showing the exact same very dramatic normalization of all of these signaling abnormalities, including ERK, P38, and SMAD activation, simply by knocking out both copies of both of these candidate modifier genes. So MMP17 is a really, really interesting molecule. It is a matrix metalloproteinase. It does have catalytic activity, but it's in a unique subset of MMPs in that it's membrane bound because of a GPI anchor. Now, independent of its catalytic activity, MMP17 has shown the ability to transactivate the epidermal growth factor receptor, and that occurs in a ligand-independent manner. It does not require the EGFR ligand. Activation of EGFR goes on to activate the RAS, RAF, MEK, ERK signaling cascade. So the signaling abnormalities that I've showed you would make sense with this. When we returned to our Marfan mice and looked at activation or phosphorylation of the EGF receptor, you can see that it's uniquely elevated in the predisposed 129 Marfan mice, but not in the black 6 Marfan mice. You can see that all you have to do now is knock out both copies of MMP17 to fully normalize EGFR activation. So this suggests that this is a MMP17 specific abnormal event in Marfan mice. On this basis, we went on to treat the 129 Marfan mice with Erlotinib, a selective FDA-approved EGF receptor antagonist, and found that this was sufficient to fully normalize aortic root growth rate in both male and female Marfan mice, and led to dramatic normalization of EGFR, PKC, and ERK activation. So really, through an extension of a modifier study, appreciation of a new therapeutic strategy for Marfan mice. So at this point, I'm going to switch gears and talk about a different phenotype that you're all familiar with, vascular Ehlers-Danlos syndrome. As you know, this is caused by heterozygous mutations in the COL3A1 gene. Unlike Marfan syndrome or Loeys-Dietz syndrome, this condition is not associated with a high frequency of aortic root aneurysms, but instead shows a strong predisposition for aneurysms or dissections without aneurysms or spontaneous vascular rupture throughout the arterial tree. So these are published information, so I'll go through this quickly. We created 2 knock-in mouse models of vascular EDS, both with glycine substitutions in the collagenous domain. One mouse was designed with the intent of making a mild mouse model, and this is what we obtained. These mice show a median survival of 400 days. And all death that we observe is due to documented arterial rupture, most often the descending thoracic aorta. We made a more severe or mutation predicted to lead to a more severe phenotype. And again, that's exactly what we got, with a median survival of 40 days. Again, all death due to arterial rupture. And again, most often the descending thoracic aorta. These mice do not show particularly dramatic histologic abnormalities, as is also described for people with vascular EDS. There's some irregularity of elastic fibers, occasional elastic fiber breaks. The aortic wall does look a little bit thin. There's slightly less collagen within the aortic wall, as shown by both trichrome staining, the blue stain, also with Picker Sirius red staining, and that's shown quantitatively on the right. So we went on to try to understand vascular EDS by, again, using an unbiased method by doing RNA sequencing of the predisposed, not yet enlarged, not yet dissected descending thoracic aorta. And we found that there were many genes that were predictably dysregulated, either upregulated or downregulated in vascular EDS mice compared to their wild type litter mates. When we asked computer algorithms to tell us what upstream pathways were most likely causing these gene expression abnormalities, there were 2 very clear genome-wide significant answers, and it was suggesting that there was involvement of a G protein coupled receptor, most likely involving a G sub Q alpha subunit of the receptor that signals through PLC, IP3, PKC, and ERK. And the other clear signal was that PKC and ERK signaling is dysregulated in vascular EDS. So we went on to ask, well, is this just a marker disease or is this driving disease? By treating the mice with either a PKC inhibitor such as robuxastorin or a MEK inhibitor that prevents ERK activation such as cobimetinib. Again, these are published, but we saw a very dramatic rescue from vascular death with treatment with either a PKC or an ERK inhibitor. And this protection associated with strong direct evidence for suppression of both abnormal PKC and ERK activation. Interestingly, the protected mice, the mice that were dramatically protected, did not show any histologic changes in the aortic wall. They still showed a decrease in wall thickness. They still showed occasional elastic fiber breaks. They still showed decreased collagen content. So this is not a mechanical mechanism for protection. Instead, these mice continue to have an abnormal collagen deficient aortic wall, and yet they are protected from the catastrophic outcome of aortic dissection or rupture. So we were interested in trying to learn what are the upstream drivers. As I mentioned, G protein coupled receptors were predicted to be involved, but tyrosine kinase receptors can also initiate the same signaling cascade. When we treated these mice with broad spectrum tyrosine kinase inhibitors, it did not lead to any improvement in survival, so that didn't appear to be relevant. We treated them with GPCR inhibitors such as the angiotensin II type 1 receptor inhibitor losartan. We didn't see that altered survival. Thrombin receptor is also a G sub Q GPCR. We didn't see evidence that made a difference. But when we inhibited the endothelin receptor, another GPCR with a G sub Q subunit, we saw that there was strong evidence for protection. In work that I'll go on to describe, but some of it already published, we know that there are other GPCRs with G sub Q subunits that can feed into this, including the oxytocin receptor. So the answer was that we were beginning to chip away at the pathogenic sequence. This led to the hypothesis that hydralazine might be protective, because hydralazine works at least in part by blocking IP3-mediated PKC activation. And initially, this looked strikingly protective. We were really excited to see by 40 days of age, when almost half of the untreated mice are already dead, we had 100% survival, but then saw that that death due to aortic rupture ramped up at about 45 days of age. Around the time of sexual maturity, we found that this was much more dramatic in male vascular EDS mice than it was in females, leading us to the hypothesis that androgens might be involved. We went on to show that use of hydralazine to prevent early death in combination with the androgen receptor blocker bicalutamide then led to sustained protection through sexual maturity and beyond. We were able to show that this could be mimicked by the diuretic spironolactone that also has significant anti-androgen effects, but are better tolerated in both young men and women. This was perhaps the second most surprising result that I had in my career, and that's what we observed when we bred our vascular EDS mutations onto an ultra-pure 129 background. So everything we've talked about has been the performance in black 6 mice shown in red. we found that when we put either the mild or the severe mutation on an ultra-pure 129 background, suddenly we saw complete survival going out to a full mouse lifespan. So these mice still had vascular EDS mutation, including a very severe mutation. As I'll show you, they still had collagen deficiency in the aortic wall. They had elastic fiber breaks. They had reduced collagen content. They had thin aortic walls. And yet they're fully protected. It could not be explained by any differences in blood pressure between the black 6 and 129 mice. And even on the protected 129 strain, if you took out aortic rings and measured their failure stress, you saw that they remained biomechanically weak, as weak as they were in the predisposed black 6 background. And yet these mice were able to survive a full lifetime. They were able to get pregnant without vascular compromise. Really, the most remarkable level of protection, suggesting that nature had found a strategy to protect from a vascular EDS mutation and type 3 collagen deficiency. So when we looked at the ultra-pure black 6 background, we saw, as I mentioned, most of the mice were dying before about 75 days of age. There were some outliers. If you looked at the ultra-pure 129 background, we saw survival up to 200 days and beyond, again, with very occasional outliers. So we did our old trick of intercrossing the mice once again, many intercrosses to generate recombination, then bred in the severe vascular EDS mutation, and stratified the mixed mice into protected 129-like mixed mice, not protected or black 6 mice, like mice that were dead before 70 days of age, and then a few mice in that intermediate zone that we simply excluded from the linkage analysis. So when we went on to map, or hopefully map, the modifier, we saw that there was, again, now this time a single locus that explained the dramatic worsening of disease on the 129 background, protection, I'm sorry, worsening on the black 6 background, protection on the 129 background. And fascinatingly, it's one of the prior modifiers that we had mapped for Marfan syndrome, MAP2K6. But now the 129 background is protective rather than detrimental as it had been in Marfan syndrome. So it's suggesting different mechanisms of modification. Again, remember, we're talking about different segments of the aorta. We're talking about the risk of rupture without an aneurysm as opposed to dissection with an aneurysm. But really a very surprising but very striking result with full genome-wide significance. So we found that if you were pure black 6 at the chromosome 11 locus, you had a low rate of survival. We saw that if you were pure 129 at this locus, you had a high level of survival. And we found that if you took protected 129 mice and simply knocked out the MAP2K6 gene, you saw that you started to see death due to spontaneous vascular rupture starting from an early age. So strong evidence that now high level expression and activity on the MAP2K6 of the MAP2K6 gene is what is protecting vascular EDS mice as opposed to making Marfan mice more vulnerable. So in reading through the literature, we learned that high activity of MAP2K6 is predicted to lead to high activation of p38, which we had seen also in the aortic root of Marfan mice on the 129 background. The high level of p38 in the descending aorta is predicted to increase the activity of two protein phosphatases called PP1 and PP2A that act by deactivating PKC and ERK. So in theory, this could be an axis of protection in the 129 animals. One prediction you'd expect is that 129 mice with high MAP2K6 activity should show high levels of p38 phosphorylation in the descending aorta, and that is indeed true, as shown in both wild-type animals and in vascular EDS animals. So that part of the prediction could be validated. The second part of the prediction is that we should see high PP1 and PP2A in the descending aorta of protected 129 animals. So what Caitlin did is she made lysates of the descending aorta from both black 6 mice and 129 mice, and showed, then used an in vitro assay using those lysates that measure the activity of PP1 and PP2A. You can measure the velocity of dephosphorylation and the total amount of converted substrate. And as predicted from this model, there's high level of phosphatase activity in 129 mice compared to black 6 animals. So that part of the prediction is also validated. The final prediction is that in 129 protected animals in the descending aorta, you should see low levels of phospho-PKC and phospho-ERK. And again, that's exactly what Caitlin saw. So you can see in the predisposed black 6 animals, the vascular EDS mice have high levels of PKC and ERK activation. In the protected 129 animals, there's not even a hint of that. There's no excessive phosphorylation of PKC and pERK. So really a clear demonstration that this prediction of low PPKC and ERK in a protected vascular segment indeed was true. So we can now validate all the predictions of this model. Another prediction would be that if you treat mice with a p38 inhibitor, it should decrease the level of the phosphatases, causing increased levels of phospho-PKC and ERK, and should aggravate aortic disease. And again, that's exactly what Caitlin saw, an elevation of PKC and ERK activation if you blocked p38, and a great acceleration of death due to aortic rupture. This predisposition that you impose by p38 inhibition can be relieved by also blocking MEK, so preventing ERK activation, showing that this axis ultimately is ERK dependent in imposing the risk of vascular rupture. What's downstream of PKC and ERK? Well, we're just starting to chip away at that, but we saw that predisposed black 6 mice that with vascular EDS have high levels of expression of endothelin-1 in the vessel wall, which is not observed in the protected 129 background. We've been able to show that if you take black 6 mice with vascular EDS that have high levels of endothelin expression shown in red, and treat them with either a PKC or ERK inhibitor, you can completely eliminate this excessive expression of endothelin-1. So now we have a really quite mature view of what's going on in vascular Ehlers-Danlos syndrome. Again, relevance of the PLC, PKC, ERK axis. definition of many therapeutic strategies, including endothelin inhibitors, including oxytocin inhibitors, and data I didn't have a chance to show you, PKC and MEK inhibitors. You can see that this pathway of modification perfectly validates this pathogenic axis and these therapeutic strategies. So really a tremendous amount that's been learned from strain-specific variation. And as my final slide, I'm showing that we're early days in looking at people with vascular EDS, but in pathologic specimens from both aortic tissue and iliac artery tissue, we see concordant findings, including excessive activation of both PKC and ERK. So we got a bunch of new ideas now that were derived from unbiased approaches, leading to a number of therapeutic strategies that are being explored, either of FDA-approved drugs or drugs that are in clinical development by drug company partners. And I'd like to end by acknowledging the truly amazing people that I have the privilege of working with every day and my funding sources. I'm sorry I went a bit over, but I am happy to answer any questions.
This is just fantastic. Thank you so much, Dr. Dietz, for volunteering your time and willingness to share all of this great data. I want to just field a first question. Do you think embryologic differences between the ascending aorta, which would be preductal, and descending aorta, postductal, might contribute to some of these background differences in the area of the aorta involved in vEDS?
Yeah, it's a great question. And as people might know, that's kind of a pet hypothesis of ours that we've been exploring in multiple aortopathies. The ascending aorta, unlike the root, is populated by vascular smooth muscle cells of cardiac neural crest origin, as opposed to a second heart field origin in the root. I think that may very well be a contributor to differences. The descending thoracic aorta is populated by somatic mesoderm-derived vascular smooth muscle cells that might be more similar to what you'd expect in the root. So it's a little bit less intuitive that embryonic origin would differentiate the root and the descending thoracic aorta. But I think it's an excellent question. There's other observations, for example, the importance of notch signaling, specifically in the ascending aortic segments as opposed to the root in determining aortic size. Those are all things that we and others are currently exploring, but we don't have final answers to that yet.
Yeah, Hal, this is Alan. And the second question that's coming on the list here is, this is Rongwu Zhang from Dr. Dieter Reinhardt's lab at McGill University. May I ask, with the amelioration of the phenotypes and intracellular signaling, ECM, dilation and survival rates under experimental conditions, do you look up the phenotypes of the smooth muscle cells in the aorta? Under the experimental conditions and treatments, are they more prone to synthetic phenotype to restore the ECM or contractile ones as they are in the normal aorta?
Yeah, again, great questions. We do not see in either the vascular EDS model or in Marfan models at least early on in disease predisposition, we don't see a major phenotypic switch of vascular smooth muscle cells. They're still expressing normal levels of contractile proteins. In the Marfan mice, later in their pathogenesis, you see a more synthetic phenotype with lots of collagen expression, still relatively preserved smooth muscle contractile protein expression. So, we're not getting a clear signal that phenotypic switch is the major determinant of disease, at least in these conditions. And that's not seen in the vascular EDS mice. The the contractile protein expression and contractility of smooth muscle cells appears to be perfectly normal.
Right. The next question starts as a statement and then kind of asks if you have any thoughts about this. Perhaps the dosage of black 6 allele that associated with the probability of big versus small Marfan aortas instead determines a hemodynamic feature known to affect aortic size, something like aortic regurgitation. Any thoughts?
Yeah, so we've looked at that very carefully. We looked at heart rate, we looked at blood pressure, but we also do very careful echocardiograms of all of these mice, including color Doppler. We're very experienced at assessing valve function in these mice. There is virtually no evidence of aortic regurgitation. There is not heart enlargement, as you might expect with a volume overload. So we really don't see any hemodynamic parameter that would explain that. Yeah, I understand the concept, though, that it might be only mice with some added predisposing factor like valve dysfunction that would show the big aorta, but we're just not seeing any evidence of that. It's a good idea, though.
So the next question was posted from Dieter Reinhardt. Given these striking differences between laboratory animal strains, is it time to start experiments with wild mice to represent the heterogeneity in humans?
Yeah, it's a great question. I don't know if other folks have any experience dealing with wild mice, but it's no fun at all. You know, you'd have a really hard time trying to control for genetic background differences even among wild mice. You know, there would likely be alleles that would be selected for within your colony because of increased breeding efficiency or all sorts of other reasons. So very quickly, even within your own colony, you'd have substrains of the mice. So, wild mice are also very nasty. They bite and do all sorts of bad things. So, and no, you know, no sequencing available. You know, so it would not be something that I would go to casually.
Yeah, actually, could I ask a question maybe at the other extreme, because you started off with the discussion of the J from Jackson strain and the N from Taconic these days, and you know, even though there's a mild change there, at least that one paper described a very dramatic difference between the two. And I guess that paper was surprising to us, frankly, because the J strain data there was way off all the published data. But those mice came from Germany, that vendor in Germany, and I guess it kind of provokes the whole bigger discussion of, you know, obviously Jackson's very aware that there's drift over time. And so if we all start breeding our own mice in our own labs, we're probably going to get a lot of drift within our own labs, potentially. Yeah. And I don't really know how to do anything about it other than make people aware that there could be differences.
So great question. You know, what we do is we never breed to, you know, when we're maintaining a colony, we never breed to other mice in our colony. We buy new breeders from Jackson every time. So they are constantly quality controlling the purity of their strains through very comprehensive genotyping. And, you know, that's the way that we avoid having drift within our colony. Every breeding goes back to a breeder that we bought directly from either Jackson or Taconic or whatever the source is.
That's a very rigorous approach.
And if it's okay, just one more question. 129 has 2 renin genes, and the C57 Black 6J has only 1 renin gene. Any evidence that the differences in aortic pathology is relevant to the renin-angiotensin regulation?
Yeah, I mean, I was not aware of that difference, but we don't, you know, what we have done is for all of our experiments constantly, we're monitoring heart rate and blood pressure. And you know, what we have not seen is any clinical evidence of a high renin state. You know, predisposition in the Marfan mice seen on the 129 background is, similarly responsive to ARBs as, were mixed mice or less back-crossed mice. So, I have no smoking gun that specifically is pointing to renin, but I also would not ever be surprised that the renin-angiotensin-aldosterone system is creating mischief. So I'd be intrigued to learn more about that.
Well, due to time constraints, I want to just wrap things up. But again, this is just such a wonderful forum. And I think if we could have more discussions like this, the whole scientific community will just benefit tremendously. And it's so generous of you to be willing to do this for us. I learned a lot, not just from your results, but even from your methodology. So thank you, Dr. Dietz.
My pleasure. And if anybody has questions or, in follow-up, or please feel free to reach out, email, call me. I'm happy to talk to anybody at any time.
Great, thanks, Hal. That was a great talk. Thank you.
Thank you.